Okay, once again, welcome. Thank you so much for joining us today. So this is the very first presentation in a series of four on the mayflies, stoneflies, and caddisflies. And before I go into the presentation, I'd like our panelists to introduce themselves, let us know who they are, where they're at, and what they do. So Jenna, would you like to start?
Good afternoon. I am Jenna Steek. I work for the Missouri Department of Conservation. I am a stream team biologist over the Southeast region, and I'm also part of the volunteer water quality monitoring training group.
Okay, Barry.
Yes, I am Barry Polton with the U.S. Geological Survey in Columbia, Missouri. I work at the Columbia Environmental Research Center as a research biologist and have a background in aquatic entomology. And I've worked a lot in the bioassessment and stream assessment programs and in other water quality related projects in USGS.
Kat?
I'm Kat Lackman. I'm stream team biologist for the Missouri Department of Conservation, and I cover the Ozark regions, just kind of helping out with tech support today. One little caveat on the chat that'll kind of help us out to field any questions. If you could do that when you select your chat and send it to all panelists, that way all of us see it and could give everybody a chance to kind of cover that question for you, that would be awesome.
David?
Hi, I'm David Bowles. I recently retired from the U.S. National Park Service. My background is very similar to that of Barry Polton, being an aquatic entomologist / ecologist, but my expertise has historically been on ecology and systematics of Caddisflies or Trichoptera.
Randy?
I'm Darver. I currently work for the Missouri Department of Natural Resources in the Environmental Services Program. And currently it's as a volunteer water quality monitoring coordinator. And previously I had worked as the bioassessment unit chief and helped develop the bioassessment program for Missouri.
Okay. And we'll have Sam here for a short time before he's got a scoot. But Sam, would you like to introduce yourself?
Yeah, I'm the assistant for the stream team program, specifically the volunteer water quality monitoring program part of it.
Great, thank you. And so all the panelists here are going to help, answer your questions or direct them to me. And all the panelists, you're welcome to interrupt me if I do miss something or go a little bit too far ahead on an issue. So I'm going to go ahead and share my screen. And we'll get going. And I may or may not see the chat, so we'll see if it pops up and I can try to address something. I'll try to do that, but otherwise I'll try to pause as well. And let's go full screen. All right, can everybody see that okay? Okay, great. All right, well, again, welcome. This is just an introduction to EPTs, Epeheroptera, Plecoptera, and Trichoptera, or the mayflies, stoneflies, and caddisflies. This is a little bit different doing it virtually rather than in the classroom, but the great thing about this is that we have so many more participants that can join in today. Otherwise, if we'd been doing this in the classroom, we would have limited the number of participants to about 15 to 20 because we actually use microscopes or dissecting scopes in actual specimens. But I'm very pleased and floored by the response that we had for registrations for today. So this will be just kind of a precursor for what's to come in the rest of the series. Move on. There we go. And this is brought to you by the Missouri Stream Team Academy. We call this a creek course in the virtual format, but typically an in-person workshop would be an academy workshop. If you're not familiar with the Stream Team program, visit us at mostreamteam.org. And for those of you who have joined us that aren't Stream Team members, that's great. We definitely appreciate your presence, but feel free to explore more about the program because it's not just education, it's also stewardship, water quality monitoring, and learning how to advocate for clean stream resources. You can also email us at streamteam@mdc.mo.gov or check out our Facebook page. And all of these presentations will be recorded and put onto the Stream Team YouTube page as well. Okay, so today, again, this is just the first in a series of 4 online presentations over the next 2 weeks. We're really going to focus a lot on the ecology, distribution, classification, and morphology of these 3 orders of mayfly, stoneflies, and caddisflies. And again, this is open to anyone who's interested. And if you haven't signed up for the following presentations, please do. So what are we going to do today? Well, in a bugs world, it'd be a little bit different, but really we just want a greater understanding and appreciation for this particular group of insects. And there's a reason why we're focusing on this particular group, because they are very strong and very sensitive water quality indicators. And also, we'll touch on this a little bit, being able to use visual and written dichotomous keys. And I'll go into a little more detail about that, on how to use the key, and then a lot of the information that will go into a key that can be kind of a foreign language when you're first starting. But again, this is kind of just to set the stage for the subsequent presentations that will come ahead. But also really to recognize the really unique adaptations that these organisms have and their ecological roles. They really are kind of that keystone community within the aquatic environment. Okay, so why are they important? Again, there's a reason why we're here today. Of course, there are various tolerances to pollution, and that can vary even within those sensitive orders as well, surprisingly. And the very important link in the aquatic food web. These organisms, along with all the other aquatic macroinvertebrates, have a really important role in helping to convert vegetative matter into animal tissue to move up the food chain. And so without these organisms in a stream or even a lake or a pond environment, you're not going to see that higher trophic level of fish and other predators or animals that might consume them. So again, very important, kind of that keystone community. And the fact that they're relatively immobile was important because when we have toxic types of pollution events, a fish has the ability to move up and downstream and maybe escape something toxic. But because they have such limited mobility on the stream bottom or the benthic zone of the stream, they really don't have the ability to escape when something toxic happens. And because of their very long life cycles, we're going to focus mostly on the larval or nymph stage of these organisms because of that long-term indication. They have very long life cycles and spend 99% of their life in that immature stage. Okay, so a little bit about taxonomic classification. So we'll have 3 different types of classification that we'll discuss. And taxonomy is just simply identifying things based on the relationships that they have with each other, usually by morphology and how they look alike. And we use a lot of descriptive language in Latin. So even if you've never taken Latin before, a lot of times it's pretty easy to figure out kind of what these descriptive names mean. And if you've been through our volunteer water quality monitoring program, at least in the introductory and level 1, portions, we typically on our field protocol for collecting data typically go to about the order level. We do go to family in some instances, but only to where we can actually identify them in the field and get that water quality rating in the field. However, in the lab, a lot of times we do try to identify these things to species if we possibly can. And if you're looking at the hierarchy of biological classification, I would actually flip this pyramid upside down because the fatter part of the pyramid is a high classification, which typically means that is going to include more types of organisms. So your order of mayflies is going to have all the families within it. Your class of insecta is going to have the mayflies, stoneflies, and caddisflies. phylum, kingdom, and so on are going to be more inclusive. So they're a higher taxa level, and then the lowest taxa level is at the species level. Okay, in addition to taxonomy, there's also feeding and behavioral classification. So all of these are tied together in determining their sensitivity to pollution. And functional feeding groups are simply groups based on how they feed. And some pretty common examples, we're all pretty familiar with predators. And there's even different types of predators within that category, which are like piercers. Those are the true bugs, or the Hemiptera. They actually have little pokey mouths so that they can actually suck the fluid or juices out of another organism. So those you have to be careful with. You don't want to handle them because they'll bite you. And then you have engulfers, like dragonflies, that actually have, the ability to grab their food and take it to their mouth and engulf the entire thing. So I think most of us are probably pretty familiar with dragonflies. Omnivores are really important because that energy transfer within the aquatic ecosystem, and crayfish is a really perfect example because they eat a little bit of everything and everything eats them. So crayfish, they're a whole workshop on their own, but they're extremely important. Also, one of those keystone species within the aquatic environment too. So crayfish are really important. And then we have collectors such as caddisflies. Caddisflies fall under several different of these feeding categories as well. You have a little bit of everything in that caddisfly order, but many of them are collectors, and that just simply means that they're collecting food from the flow of the stream. Especially our net spinning type of caddisflies, where they actually build a net with the silk that they produce. They can actually pick bits and pieces of their food. That could be bacteria, diatoms, pieces of algae. And then put their energy in repairing that net in order to feed again. And so they're a really good collector group. And then scrapers and grazers, snails are definitely a very good example of that. And if you have the patience to sit and watch a snail, they do move very slowly, but they leave a trail of clean rock behind them as they scrape the algae off the rocks. And we have a lot of those types in the mayfly and caddisfly and in the stonefly groups as well. So even within the mayflies, stoneflies, and caddisflies, they're not necessarily one type of feeding classification, but often many different kinds. Now, shredders are extremely important, and stoneflies, I can't emphasize enough how important stoneflies are, and then being so sensitive to pollution, they're usually the first group of insects impacted when a stream is impaired. But shredding just simply means taking that coarse particulate organic material like leaves that fall into the stream and shredding it up and making it more fine particulate organic matter for that material to move downstream for the collectors and the grazers to consume. So again, everything is connected, even within the feeding classifications. And then parasites, we won't go into much detail. Water mites are a good example, but really only 1 or 2 parts of their life cycle are actually parasitic, and it's on other invertebrates and not on humans. But everybody has a place within the aquatic environment that's important. So on top of feeding classification, we also have behavioral designations, and that's just simply how an invertebrate moves throughout its environment. And again, within the groups that we're talking about, these are different among those groups, but a good example would be clingers. A type of spiny crawler mayfly or a flathead mayfly would qualify as a clinger. And you kind of know you got a clinger because that body's going to be flat. And it's flat because it really wants to hold on to that rock that the water is rushing over. And so you really kind of see that type of character, prevalent in how it's going to move about its environment. So clingers, definitely mayflies, a lot of stoneflies are definitely clingers. And then we have climbers, such as damselflies, or the Zygospora suborder within the Odonata. And they are really good at climbing up roots along the sides of a stream, and it's necessary for them to do so, not just for feeding, but also for emergence. So very good crawlers and climbers and able to get out of the stream in order to emerge into adulthood. And then you have the dragonflies, which is the Anisoptera suborder of Odonata, so the cousin of the damselfly. They're really good sprawlers. They can just kind of anchor themselves on the bottom of the stream, and because they're really good sight predators, they can actually wait for food to come to them, well camouflaged in their environment, and just grab their food as it comes to them. Or, climb and walk across the bottom. They're not really great swimmers, but they do have pretty strong legs. And then you have burrowers, and a burrowing mayfly in the family Ephemeridae is a really good example. And burrowers, they're not just necessarily in the rocky type of substrate, but also in the sandy and silty types of substrates. And they have special structures on their body in order to be able to dig into that substrate where they can be safe from predators. And then you have the swimmers, where they are a little bit more mobile. Again, not as much as a fish would be, but they can swim very quickly, at least up and down the water column or within a small area, and are minnow-like mayflies like the family Betidae and the Isonychiidae. They are very good swimmers, very strong swimmers, and you can see by their shape and their form when you come to the mayfly presentation on what gives them the strength to be able to swim. And then you have divers, and that's essentially going to be most of your aquatic beetles, especially the adults that do actually breathe air and require to get an air bubble in order to move up and down the water column. So all of these things are connected. Their morphology, their feeding, and the behavior helps to determine how they function within the environment and their sensitivity at oftentimes as well. Now, in order to measure that, metrics is a way to quantify that community structure or biological integrity. What's going to live there? So this is important, and we actually do this in the Volunteer Water Quality Monitoring Program. It's a little bit different than what you do at the genus and species level. Here in Missouri, and it is different across different states depending on the types of organisms that you have present. But the tolerance index is essentially a scale for showing the quality of the environment by indicating what's living there. So here in Missouri, taxa are assigned tolerance values from 0 to 10, and 0 being the most sensitive and 10 being the most tolerant. Now, it's kind of the opposite of the stream team protocol, where we actually designate a higher number for our sensitive organisms and a lower number as they become more tolerant. But that's because that is strictly a field-based type of protocol. And what's used here is these are samples taken back to the lab, looked at under a dissecting scope, and identified to genus and species in many cases. So common metrics at the family level can be anywhere from just the percentage of EPTs that you find in a stream sample compared to everything else. The taxa richness, so the amount of different types of taxa that you have available. And then there's one that was developed out of Wisconsin that I like to use, is the Hilsenhoff Family Biotic Index. And they use the same scale, but at the family level. But here in Missouri, it goes all the way to species level. And I've kind of seen some stuff pop in on the chat. Are there any questions?
Yeah, there's one, Amy.
Mm-hmm.
What is behavior designation of different types of caddisflies?
It's all over the place. So, and that's something David will talk about in his, and I'm sure he'll address it in some way in the Trichoptera presentation on March 30th. So there are several different types of behavioral designations for caddisflies. So for the most part, here, they're kind of be in that clinger, sprawler type of category more than likely, unless David has another suggestion.
Yeah, they're variable depending on whether they're grazers and case builders or shredders or summer predators. So their behaviors are very different, even within a family.
Yeah, so even throughout these groups, they're going to be kind of all over the place. So these were just a couple of examples just to kind of show you maybe what kind of body structure these types of behavior designations might have. All right, so we're all about Epeheroptera, the mayflies. And again, we're focusing on the nymph life stage. So that's the immature life stage of something that is hemimetabolous or with an incomplete metamorphosis. And that simply means that they continue to grow as a nymph and molt. And in between those molts, they're called instars. And then they will emerge directly into that adult stage. And Plecoptera are the stoneflies, and they're the same kind of life cycle of hemimetabolous and having that incomplete metamorphosis where they emerge directly into the adult. Of course, their immature stage is 99% of their life cycle. For a mayfly, their adult stage, and they actually have two, a subadult and an adult. They're unique, and that's the only insect that actually molts again after it emerges. And so you have the subimago and the imago. But that whole system, that whole part of their life cycle might be a day or two. And then a stonefly will also live about a year mostly, but up to 3 or 4, depending on the type of group. And of course, you'll learn more about stoneflies in depth when Barry does his presentation, and that will be on March 25th. But their adult also doesn't live for very long. They might live a week or 2 at best, and they don't go very far from where they've emerged. And then you have the Trichoptera, which of course are the caddisflies. And these have a full life cycle or holometabolous life cycle complete, and that just means that they have that larva stage in between the immature and the adult. So mostly we'll call mayflies and stoneflies nymphs in their immature stage, or naiads, and then the Trichopteras and the caddisflies are actual true larvae because they have that pupa stage. Okay, so you notice at the end of each of these order names is the word Ptera. And if you guys are, it's one of those Latin words, but after what we've been talking about, what does Terra mean? And you can go ahead and throw that in the chat, and I've got a little hint here of a pterodactyl. And I can see some answers coming in. Yes, very good. So that means they have wings. Yeah, so they're winged. They can fly. Now, and then if we put those Latin words together, Ephemeroptera, any ideas what ephemera means? And if you've, you know, sat by an ephemeral stream, that might give a hint. Okay, good. Yes, short wing, mainly probably because of their such a short lifespan and their short flight. They really don't go very far either. They don't have the power to fly, so they can really only fly short distances, but I think it probably mostly applies to their lifespan. So good. What about Plecoptera? Any ideas what pleco means? This one's a little tougher. Two. Any other guesses? Okay, yep, this is a tougher one. And I actually found 3 different types of descriptions for pleco, and I think folded might be the most accurate because they do hold their wings folded over the back of their abdomen. But also breathing and gilled makes sense because of that breathing action in the water and how sensitive they are to oxygen. And many of them do actually have gills at the point of attachment of their legs, or as we like to call them, the hairy armpits. Okay, Trichoptera. What would tricho mean? And it may not be as intuitive as it seems. Any guesses? We have 3-tailed, hair, threefold. Yeah, this one's deceiving, but yes, hairy wing is correct. And you think of tri as three, so tricho, but I think the whole Trichoptera put together, yes, hairy wing is the definition. And actually it makes sense if you've seen an adult caddisfly, they hold their wings roof-like over their back, but they are very moth-like, and they have those really tiny hairs on their wings. And so it is still a pretty telling and appropriate name for caddisfly. Okay, any questions on that? Okay, so we'll move on to using a dichotomous key. Now this is going to trick you because tricho meant hairy, but dichotomous does mean two. So simply a dichotomous key, and this is the tool and the resources that taxonomists use in order to identify an organism, and it can be any kind of organism. And really all it means is that this is a key that gives you two options or two choices when you have a specimen sitting in front of you, and which of these options fits best. So we use a visual key in the Stream Team program, and this can be found on the Stream Team website. I did not include that in the original documents that you could have accessed before the presentation, but this one's easy to find on the website. And really simply, it's using a visual key in order to tell you what you might have going to the order level. So simple as starting as with a shell or no shell, legs or no legs, and then so on and so forth. And then matching your specimen the best that you can to the final options. So dichotomous key is simply two choices in order to get to the identification as best as you can. And it gets more complicated as you go into the family level, the genus level, and most definitely the species level. But there's another type of dichotomous key that is using mostly text. And so it depends on if you're a visual learner or a wordy kind of learner. I kind of need words a lot of times. And again, you have a couplet of two options to choose from. And it will take you through that whole process until you get to the best fit for the specimen that you have in front of you. And I believe this one we did send to you ahead of time, so use that as a resource. Okay. So that's just a quick rundown on using a dichotomous key, and you'll definitely get to be a lot more experienced with that with the following presentations that will come after this one. Because the rest of this one, I'm going to focus on morphology. So this is kind of a crash course in entomology that took me an entire semester to get through, and you're going to get it in about the next half an hour. So I appreciate you putting up with me here. But morphology is simply the form and structure of an organism in all of its parts. And so all of those parts have names, and it's just all it is to describe an insect. You could say he's an ant, but, well, he's an ant, right? So it takes some descriptive work to get to an ant if you don't know one immediately off the bat. Okay, so some of the morphological terms that you're going to come across will have prefixes. And so these are words that are pretty intuitive. And so you would know that something that's at the fore end of something would be at the front. The three segments behind the head begin with pro, meso, and meta. And I'll go into more detail, and you'll get to see this on an actual virtual organism. Now, in addition to those prefixes, you'll have descriptive terms on what part of the body you might find a feature. And so ventral means that it's on the underside or the belly, underneath. Okay? Dorsal is the top side or the back. Lateral is on the sides. And then you can mix words together, right? Because most of these are Latin and they're descriptive terms. So dorsolateral means at the side of the upper surface, ventrolateral at the side of the lower surface. So you'll see these words a lot. And then you have additional descriptive terms that is even more specific to where you'll find a feature on the body. And so proximal, I'm going to move my little panel here. Proximal is just concerning the basal part of an appendage closest to the body. So like your armpit is proximal to your torso. Apical, at the very tip or the apex of an appendage, furthest away from the body. Marginal, pertaining to the edge or outside border of an appendage. And then terminal, at the very end of a body, or distal. So those words can be kind of interchangeable. Okay, so for example, we're looking at a drawing of a mayfly, and we see the top side is the dorsal, the bottom side is the ventral. The very tip of the leg is the distal part of the leg, and then that basal segment would be that first attached segment to the thorax. And then lateral courses to the side, posterior is at the edge or the rear, and anterior is at the front or the head. And the median simply just means middle. Okay, any questions about morphological terms? Because I'm seeing stuff pop in, but I can't get to them fast enough. Sounds
Those
like
must
our
be coming.
panelists are.
Those must be coming straight to you, Amy, because I'm not seeing anything come in the chat.
Okay. Yeah, make sure that you're addressing all panelists because I may not be able to see your chat as it comes in. But at the very end, I'll go back and see if there are any questions that didn't get addressed. Okay, so most people remember that there are 3 basic body sections to an insect, and we know it as having the head, a thorax, and an abdomen. And for this particular group of organisms, this is true, where they do have a true head, a thorax, and an abdomen. For those incomplete metamorphosis type organisms like the mayflies and the stoneflies, they're going to have wing pads as well. And so each section of the thorax, the pro, meso, and meta, has a segment for the attachment of each pair of legs. And then the mesothorax and the metathorax have points of attachment for the wing pads on those hemimetabolous nymphs. Okay. The abdomen can be extremely variable in all insect groups. Thankfully, among these 3 groups, the number of abdominal segments is going to be 10, although in other groups that number is variable. But we know for these, for purposes, being able to count the number of segments on the abdomen is going to be really helpful, especially on caddisflies and on mayflies as well. Okay, if we take a look at the insect head, and this is not an EPT, I believe it's just a rendition of possibly a grasshopper. But your head parts are all going to be the same. But maybe with varying features on them, where those descriptive terms will come in handy in pointing you in the right direction where those features. So, kind of neat here is a distal cleavage. These cleavage points are essentially where the insect, when it molts, is going to split. So they'll split open on the head and the thorax and then emerge anew as a new instar as they grow. The frons is essentially the face or the forehead. The clypeus would kind of be the equivalent of a nose, but flat. And then you'll see the word frontoclypea, clypeus, or frontoclypeal sulcus. So again, these descriptive terms just mean this is the division between the frons and the clypeus, which is essentially the nose. Okay, and then the labrum is the upper lip, and is typically one piece, but is essentially the upper lip, and then mandibles, which are the hardened tooth-like structures on the mouth. And we'll go into a little more detail on the mouth parts because this gets kind of complicated, but it gets really important when you're looking at stoneflies and mayflies. Mayflies, especially to the genus level, and then stoneflies at the family level. But on a mature specimen, it's pretty easy to determine the different choices key in that dichotomous key on the way that they're shaped. All right, so the lower lip, and this is kind of where I get a little goofy, And the upper lip is the labrum, so it's kind of one piece, but the lower lip, the labia, it's a little more complicated. And I always talk about how monsters and aliens kind of these creatures that a lot of our horror film creators make comes from the insect world in a lot of ways. So if we actually pull that lower lip down on the ledge that you see there, the mentum, the prementum and the submentum are going to be the attachment to the mouth. And then you're going to have little labial palps on the outside of your bottom lip. And then your glossy and your paraglossy on the outside and your glossy on the inside. And they're going to be shaped differently depending on the group of stonefly. It's a really important mouth part. And then if you look at the side, so now we've got the lower stuff going on, and then we got the upper stuff going on. And your maxillary palps are going to be fully to the outside. And then I wouldn't worry too much about the other parts unless you're going to species with stoneflies, but then your mandibles will be there. So your maxillary palps kind of help sensory to maybe guide food into the mouth, while your labial pipes just kind of shove it in. So looking at it from the side, again, your clypeus is kind of your nose, your labrum is your upper lip, your mandible is that hardened chitinous or tooth-like structure, and the maxilla is what includes the maxillary palps and the mandibles, and then your lower lip on the labium. Okay, going a little deeper now. Again, this is going to come in really handy with your stoneflies. So again, we're looking underneath that labium there, that lower lip, and you've got your labial palps on the outside, and these are segmented, and that's going to come in handy too. And honestly, depending on the type of magnification that you're going to have, these may or may not be easy to see. And I would suggest 30x power minimum to be able to identify these, especially if you've got smaller specimens. But this does become important with stoneflies. And then again, you have your paraglossy on the outside and your glossy on the inside. Okay? If we take a look at an actual specimen, and you'll notice this is a little bit different, how the glossy and the paraglossy are kind of rounded and soft. On this guy, his paraglossy and his glossy are a little more angular, more pointed. But you can see those maxillary palps, maxilla including the mandible, and then the labial palp, the glossy, and the paraglossy. And then your prementum, mentum, and submentum. The mentum on this particular group of animals isn't as important, but if you're ever wanting to identify midge larvae, it's all about the mentum. And it's a really tough taxonomic group to crack because the mentum is a different shape in every genus, and you have to actually smash them down and put them on a slide in order to identify them. I know I've struggled with that. Okay, if we look at the head on the dorsal side, we can see again that maxillary palp hanging out on the side. It's kind of that sensory to help kind of guide stuff along. Here we have the upper lip or the labrum, and it's highly modified on this particular specimen, and you'll learn more about them in its talk on March 30th. And again, you'll kind of see that division between the front of the head or the face and that that line between the forehead and where the mouth begins. And it's highly modified in this particular specimen, but actually that will identify this particular guy to genus, which is pretty cool. And then the occiput is just the entire head capsule. The cervix is the soft tissue between the head and the first segment behind the head, which is the pro. All right. What do we have here? What do you guys think this is based on its type of structure? You can throw that in the chat. And the head is cut off. See, it's an ant, right? Just like the cartoon said, but it's a little more complicated than that, as you can see. And this is much more complicated than the groups that we're looking at, but I thought I'd throw it in there just to show you that we have the 3 different sections of the thorax, the pro, the meso, and the meta. And then on an ant's a little different. Their abdomen actually starts where it looks like they're still part of the thorax, but they have this constriction in between. So that can become complicated. Identifying ants is also quite difficult. But I will show you a little bit more in detail on something that'll be a little bit more simple, I think, to be able to identify when you have it in your hand. Okay, so I mentioned that the first segment behind the head is the pro, and that'd be the prothorax. The second segment is the mesothorax, and the third segment is the metathorax when you're looking at the thorax as a whole. However, if you're looking at the dorsal part of the thorax, this is actually called the notum. So each segment behind the head is going to be the pronotum, the mesonotum, and the metanotum. That's when you're looking at it from the dorsal side, okay? And then the forewing pad, especially on a mayfly and a stonefly, you'll see where that forewing pad is on the mesonotum, and the hind wing pad, especially on stoneflies, are a lot easier to see on the metanotum. And this is a mayfly here. And you see those tusks. You might be able to figure out what kind of habitat this particular guy lives in based on those types of mouth parts. Okay, so now we flipped a little guy over. We went from a mayfly to a stonefly. But the bottom part of the thorax, you still have the pro, meso, and meta, but it's also the sternum. So when you're looking for a structure or a gill structure on the prosternum, you're going to look underneath on the ventral side. So again, the prosternum, the mesosternum, and the metasternum. Now, we're going to look at the side. And each side of the thorax is actually called the pleuron. So you have the propleuron, the mesopleuron, and the metapleuron. And you'll notice on this guy, they've got kind of hardened sclerites or structures here that are hard-plated or chitinized. And there could be a special structure on the mesonotum or the mesopleuron. And I use pleuron is a lot more common when you're looking at like riffle beetle larvae. Okay, and then we've got a good look at the leg on this guy, and this is a mayfly. And we'll take a little bit closer look at the leg here on the next slide. But again, so notum, sternum, pleuron on the thorax. Okay, and on an insect leg, the coxa is kind of like the ball of your shoulder attached to part of the thorax there. They have another little attachment between the coxa and the strong part of the leg called the trochanter. And this becomes really important in a lot of the caddisfly groups. So that could have a special structure or shape to it. So that's something to look for, and it's usually pretty easy to see. Again, the femur, like we've got, the strong part of the leg, the tibia, kind of like where our calves are located. And then instead of fingers and toes, they have tarsi, and each individual segment is a tarsus. Okay. And the number of these can vary from group to group, but this is also another important thing to keep in mind. You'll notice when you're going through the stonefly key as well. And then sometimes, especially for those burrowing type of organisms, you might see tibial spurs to help them anchor themselves into silt or mud habitats. And then claws. On a mayfly, you have one claw per the end of each leg, and on stoneflies, you have two. Okay, again, the abdomen can vary in the number of segments, but we are lucky that this group is pretty consistent and has 10 abdominal segments, and they are numbered from 1 to 10, number 1 being the first one behind the metathorax, and then you can count each segment 7, 8, 9, 10, all the way to the end. Okay, now remember the thorax, the top of the thorax was called the notum. Well, the top of the abdomen is called the tergum. So each individual segment, when you're looking at it from the top, is a tergite. Okay, but on the bottom, it is like the thorax, where it is still the sternum on the underside of the abdomen or the belly. And then each individual segment is a sternite. And then again, the side is still the pleuron, and each individual segment is a pleurite. This is really important because there might be special structures on, sternite number 9 or tergite number 9. Being able to count those segments and identify where they are will help you run through that key pretty quickly. Okay, so the abdomen is the most variable portion of the body. It can be a distinguishing feature, though, when you're looking at something. Say you accidentally cut its head off and you're not really sure. But the trick is just to kind of get that search image in what you're looking at, and that will take you to order right away, usually pretty quickly. Things to look for: are there hair-like cerci or tails, and how many are there? Because typically mayflies may have 3, but they can also have 2. So that might take you through the key a little bit further. But the main character that we want you to remember about mayflies, which if you haven't been through our VWQM workshops, I hadn't mentioned that yet in this particular presentation, but the gills along the side of the abdomen are the most important feature of a mayfly nymph. But the 3 tails are going to be the first thing that you notice, but sometimes they only have 2, so they can trick you. But stoneflies always have 2 tails. Now, if those tails are present, are they covered in tiny hairs? Are they cetaceous, like our little tails, our legs might be, some of us, and then, or are they bare, glabrous, and like some of our legs might be? So just something to kind of keep in mind, to look for hairs, look for tails, how they're shaped, how long they are. These are things that are, you kind of want in the back of your head when you're reading through a key that you can identify quickly when you're looking at a specimen. And then, are there prolegs? And we've seen the prolegs on caddisflies. They do have 2 additional, not walking legs, but prolegs that help to anchor them to their substrate, that have hooks on the end of them. So are those prolegs present? Are they long, or are they up against the side of the edge of the body, the end of the body? And then, where are the claws? Are they big? Are they hooked? Do they have teeth on them? These are things that we'll look at in the caddisfly key. And again, the number of abdominal segments, you count them from 1 to 10, where there might be hard plates or soft folds, gills, or lateral appendages. So again, it's just getting that, trying to understand, an aquatic insect body is going to have all these different kinds of features and more. Okay, so quiz time. This is really easy. But what do we have here? So you only have 3 options. Is it a mayfly, a stonefly, or a caddisfly, based on what we see on the abdomen? And go ahead and throw that in the chat. And remember to reply to all panelists. Okay, good. It is a caddisfly. Okay, number 2, what do we have here? We can narrow it down a little bit more. Good, yes, it is a mayfly. And then out of process of elimination, who will we have here for number 3? Good, stonefly. Very good. And again, stoneflies, I didn't mention it before, but they'll never have gills along the abdomen. Mayflies will always have gills along the sides of the abdomen. And caddisflies will have gills sometimes underneath on the ventral side of the abdomen, but it's a pretty limited group of caddisflies that do. Okay, and just a little bit more fun. Being a taxonomist, I've had some taxonomy experience. It is a challenge. It's fun, but you always wonder about that one little critter that just doesn't quite fit the mix. And and whether they're trying to fool us or not. So do we have any questions on morphology? Or anything that needs to be clarified? Give you guys a second. It's a lot to take in. Especially those mouth parts. That gets really complicated because they're just kind of all over the place. Okay, for those of you who have stuck around, thank you. If you provided your full contact information when you registered, you'll be entered in a drawing to receive a copy of an introduction to the aquatic insects of North America by Merritt and Cummins. This is a really great resource if you're into taxonomy. So if you're still here, then hopefully you are to a certain degree. It goes into a lot of detail, and it's all in all of the different aquatic insects. So you get the dragonflies, mayflies, beetles, true flies. Some of the taxonomy has changed since this edition has come out, but it's still an extremely valuable resource. So one lucky participant in each of these presentations will get one of these on a random drawing. So of course, if you are chosen, I'll definitely contact you and let you know when we get those shipped out. And it might be after the end of the series, so we can kind of get them all out at one time. Okay, and with that, we want you to stay tuned for more. We've got on March 23rd at 1 p.m., Randy Sarber will be talking about mayflies, and we'll get to really dive into that key. On March 25th at 1 p.m., Barry Poulton will talk about stoneflies, and that will be really interesting because he's got extensive research history with stoneflies, really neat ecology. And then definitely stick around for March 30th at 2 p.m. To hear about caddisflies from David Bowles. Caddisflies are one of my absolute favorite organisms because they're so unique and so industrious. Because you can actually identify them by their case, almost to species level in a lot of cases. So very interesting critters, very diverse and very unique in their structure. Okay, with that, I will go back to see if I can open up that chat. And see what I missed.
Did have one just come in. Amy, do you know how quickly we will be posting the recordings?
I'll try to get it set up and processed as soon as possible. It just depends on my bandwidth and processing with YouTube. It'd probably be about a day, maybe 2 tops. And hopefully we don't have to edit anything out. I think we did okay. But let me see if there's any that are directly to me from earlier. Let's see, has to present me. Okay, all the good guesses on the quiz.
Couple of latter ones that just came in. Do you happen to have a key to Latin pronunciation you could share?
Ooh. That's a really great question. You know, I think I try to read things phonetically, and I know that I don't necessarily pronounce everything the right way. I know I've pronounced things differently than Randy does. And actually, Randy had a really good trick on those really long names where your emphasis is on the third to the last syllable. And so I've been trying to adopt that particular I guess, systematic for pronouncing some of those kind of complicated words. And Randy, if you want to chime in and have any other tips on pronouncing, I think we all do it a little bit differently because a lot of us were learning them by reading them, not hearing them from other people necessarily.
Yeah, that's just how dangerous I am at Latin. But I think that's a Latin grammar. Every time you come up with a rule, there's going to be exceptions to the rule, but that's the way I understand. You know, third to the last syllable. is the
Yes, David, I concur with Randy. That's the standard. It doesn't always work out that way, but for most cases, the third syllable is what you put emphasis on. And the other thing I would say is in Latin, in endings, that the A is silent. So you pronounce the E. So like in the family, it's a D, no A in there.
Right, like the mayfly beta D or Cenidae.
Right.
And some people say Canidae because it starts with a C, CA. But if you're not pronouncing the A, then that would be a soft C.
Yeah.
So it's a lot of trial by error, and just sounding it out the best you can. But not everybody pronounces things the same way.
I would say even among specialists that there is often disagreement about how to pronounce a certain name, and that's okay, as long as you know what you're talking about.
We got
Another question for you. Oh,
Yep.
go ahead. Go ahead, Randy.
I was going to say there's a question from Al about why has taxonomy changed?
I guess because it's an emerging science and you find new structures. And actually, Randy, you'd probably be better at answering that one.
Well, there's no doubt it does change. It's still changing. So it makes it confusing. But as people delve farther into taxonomy and looking at morphology and even genetics now and trying to, confirm the past 150 years of taxonomy with genetics, it just continues to change, and the relationships, because it's based on phylogeny or whatever, like you said, the relationships between critters, it's reestablish, I guess, the relationships as they look at further information, especially genetics nowadays.
Genetics is the game changer.
Yeah, and that's relatively recent.
Another one for you. How about are there any dissecting scopes that work well in the field?
Yeah, that's a good question. I'd like one for sure. I know we've had some ones that we've given away at some of our prizes, but I haven't played with them. So Kat, actually, you might have a better feel for that. Of course, we can't really recommend certain brands, but anything that uses natural light and a double ocular is going to be easier to use in the field than a single ocular.
I know there's been a couple that we have given away as activity prizes. They weren't technically dissecting scopes. They were more the microscope, but they were small and portable, and were able to take good pictures through them as well that were at least magnified to be able to get better glimpses at those different pieces to be able to help identify them.
And if you don't have access to a microscope, I'd also suggest just get a good old-fashioned hand lens or a loupe. Many of the characters that you will need to look at are perfectly visible with those inexpensive devices.
And they're getting cheaper and cheaper.
That's especially true. That's especially true too with the if you're trying to ID these critters down to family. I think a lot of the family characters on most of them are visible with a hand lens if you have a real good quality one.
There's a lot of attachments now for cell phones too that work pretty well, and then you can get a really good photo.
We give away a pretty good hand lens, a 10x hand lens that has a small LED light underneath of it, and that comes in handy. Lighting is always comes in handy, but the trouble with taking out a even if you could get an old Leica or something, with a 30x dissecting scopes that are kind of all metal and stuff like that they used to have a real common or whatever that wouldn't be cost too much, is number one problem is going to be lighting. Number two is, unless you euthanize things, they're not going to cooperate. Things are going to be swimming around, moving around, or whatever. And so stream side, where you got live organisms and stuff like that, it just doesn't work real well. It works a lot better if you're going to key things out to have euthanized them in alcohol or something like that. And so you can see what you want to see.
We had the question of the name of your YouTube channel. I think you can just Google Missouri Stream Team YouTube and it should come right up. I don't know if everything is 100% accessible unless we send you a direct link. Most of what we've posted is unlisted, but we can direct. I think if you Google it, you can still get to it. Yeah, as far as URLs, it's always a funky kind of, yeah. Not one you can remember.
I think I sent that one privately to Steve that asked that one. Dan has lots of questions about caddisflies.
All right.
Are the types of caddisflies found generally in the same small area, like 200 yards on the stream, or do the types of caddisflies overlap in a given area, multiple or many or more?
Yes. Is the short answer. I mean, you can find hundreds of the same type of caddisfly under the same rock, and that definitely can be true for the net spinning caddisflies, but also the case makers will clump together because they were born in the same spot, so they've decided to make their homes there. But you have caddisflies that can live in multiple habitats. Especially our case makers. Some of them will individually prefer a slower part of the stream, and maybe they like the faster water when they're littler or bigger. That's a good question, and I don't know if David might be able to to shed some light on that too.
No, I think your answer was really good. It depends.
Yeah.
It depends on the kind of stream you're looking at, but also the particular habitat. But it's not uncommon to find multiple families in a riffle habitat or multiple families in a pool habitat. If it's a degraded stream, you might only find a couple, three. So it just depends. So if it's a clean stream, expect a lot.
And yes, we will be, I believe we have all of our creek courses on the website somewhere, but if not, I think they just link right to the YouTube page.
So Dan's asking a lot of questions also about hatches, and I'm going to guess that he's into fishing, but and spring-fed streams and temperature, and I think in general that's a hard one to answer too, but generally speaking, I think a lot of what we're talking about, when their emergence, or at least their growth spurts, is temperature controlled. You could think of degree days. Maybe if you could calculate degree days or something like that, you'd have a rough estimation of when they might emerge in a particular year. I'm not saying light isn't important, because they definitely respond to light also. But the thing that's surprising about springs is that really limit the taxa that can be there. Some people think that they're kind of, anyway, that because they don't have as much temperature change and difference, it's actually fewer taxa can actually survive in that kind of situation where temperature is the key to them emerging. I don't know if I answered that very well or not, but that's as short as I can get it, I think.
Springs usually, it's been my finding that springs usually have about 1/3 or potentially less of the taxonomic richness of a nearby stream because of the thermal constancy and cold water. It's just really old. But the densities, the numbers of those organisms are often much higher in contrast. You're muted, Amy.
Aha, I did it again. Thanks. Just started blabbing. So Meredith asked if anyone has used the lenses or the magnifiers that attach to iPhones, and April has been really successful with those and taking really magnificent photos. I tried a lens attachment to my Android and I couldn't get it to fit quite right, so they kind of have to be specific to the phone. But April's taken some pretty amazing photos, so yes, they work really well. Okay, I think Randy and David got the hatching times on spring and regular streams or spring-fed streams with a relatively stable temperature. I think that was addressed.
How about, will the course touch on within the orders, the differences in pollution tolerances?
Yes. So short answer, yes. Oh no, the questions are great. I really appreciate it.
How about, why are sometimes there are lots of caddisflies in a stream, but very few mayflies or stoneflies? Why not all of the sensitive ones?
Yeah, and you might find some of those more tolerant caddisflies in large numbers and maybe not so many of the stoneflies. And that's typically the experience that I've had. And again, you know, stoneflies and mayflies can be very specialized in their feeding behavior. And so if there's not good feeding opportunities or certain types of pollution that perhaps the caddisflies can tolerate a little bit better, then that's what you might find. So that's why it's important to monitor streams over a long period of time to really get that, you know, the stream condition over a long period of time, because that can change. Your complete assemblage of macroinvertebrates can change depending on what's feeding the stream.
Yeah, you're right. You're hitting on pollution tolerances, which it could be if it's especially if it's a urbanized stream or something like that, where there's less taxa there. People might want to read about the stream continuum concept too. And that's where we're talking about the functional feeding groups and stuff like that, and the processing of matter. So you change from scrapers to maybe like filter feeders further on downstream. So, and some caddisflies are filter feeders, you know, so you kind of have to pay attention to where you are in the stream system too.
And then we have a question from Ross. How widespread are stoneflies in Missouri? Are they more rare than mayflies and caddisflies? And I think I'll kick that one to Barry.
Well, I think overall the general diversity is maybe pretty similar in the better quality streams. I've been to several streams down in the Ozarks where I was getting about the same number of taxa in all three groups, but then there's others that may be be degraded or partially degraded, and then it might shift the balance a little bit. So it all depends on when you look, and also what season of the year you go to as well. Over 75% of our stoneflies are already emerged and gone from the stream by summertime. So they're all emerging in winter and spring months. And so if you go out in the fall, you're less likely to find a lot of stonefly diversity than you are in the spring. So sometimes it shifts. And part of that is based on because of the stream permanence. Some organisms do really well in temporary streams and others don't. Some of them need to have permanent flow. So there's a big factor there, as well as the temperature regime of the stream as well. So it just varies. It depends on where you are. It depends on whether you're in the headwaters, where you might have a more closed canopy with a lot of tree cover versus something that's more open. That'll make a difference too.
And Dan also asks, do the types of caddisflies on a particular stream look similar? I don't have an answer for that one. I mean, you can have the same species, but...
Well, and he's got a couple other caveats to that too. And he was kind of referring to the adults, asking if you could look at the flying caddis and tell anything about them.
The adults are much more rigorous to identify. So to most people, they're going to look very similar, but once you start looking at genitalia, which you're not going to be able to do in the field, they're going to be much more difficult to separate as opposed to like the genus or even family levels for the larvae. But for taxonomic precision, we have to look at the adults. And I'll get at that somewhat in my talk about the adult diversity.
Yeah, and I think a lot of these questions will be addressed. So hopefully you guys will will definitely come join us for the future presentations. So I think we'll give it about 3 more minutes. There was a comment from Jack on in a recent Penn State webinar, they mentioned that streams in a forest corridor versus open are more productive due to enrichment from fallen leaves. Any comment on that? And I would say that's absolutely correct.
Are you looking for comments then?
Yeah, I mean, I think that's pretty cut and dry.
In forested ecosystems, that was the energy driver, you know, for a lot of streams. That's where energy came from, is leaf fall. I mean, it didn't contribute a lot of energy, as you can imagine. I mean, if it's strictly from leaves, but so you bump up the nutrients in a stream, and you might not change the community some, but you might get a big boost in the abundance at first. Eventually, you're going to change the community structure, but yeah, there's a lot of interesting information about that and how fast that carbon is processed in streams too. That can also change the diversity in streams. So
that's a good point.
Yeah, just to add to what Randy was saying too, there are a lot of EPTs really in every one of these 3 groups. There are a lot of species that have adapted to their life cycles to the autumn leaf fall. And so you see a lot of species that may be in the egg or small nymph stage just as the leaves are falling in the fall. And that's when they start doing their most rapid growing and feeding, is during the winter months, to take advantage of that new food source that's falling into the stream.
Okay, time for one more. Let's see. Oh, question from Bob. Are changes planned for our VWQM biological monitoring protocol to identify macros to the family or genus levels like a few other states do? And at this time, no. I believe there was some effort before I was on board, but at this point, anything we do looking at these groups at a lower taxonomic level is going to be purely educational. I don't know, but that's something we can definitely discuss. See if we can revisit that. Okay. Okay, last one, and then we should probably sign off. Ross says, I've noticed quite a bit of erosion from flooding in recent years and curious of the impact that is having. And that my response to that would be simply, it was going to take a good amount of time of collecting data over the years and seasonally to really see what those impacts might be, because erosion could have a lot of other factors involved with it, for sure. And I don't know if anybody else has a thought for that.
This is David. Yes, in some areas we've seen decreases in caddisfly diversity in streams that have a lot of upstream bank erosion because of the increased silt is hard on the filter feeders, so they tend to drop out. So in general, and it fouls gills as well. So in general, erosion its tendency to increase is a really bad thing for aquatic invertebrates.
All right, you guys. Well, thanks so much. I really was very impressed with the turnout. Thank you to all the panelists for helping to field questions. And yes, join us again next Tuesday, March 23rd at 1 o'clock for a little more in-depth presentation on mayflies. So with that, I'm going to stop recording. And again, thank you.